Welcome everyone to our channel Dentistry for Beginners. So today we are going to discuss about tooth development. Tooth germ. The tooth and its supporting structures are formed from the tooth germ. This tooth germ is divided in three parts: enamel organ, dental papilla, dental follicle. Enamel organ is the ectodermal component and it forms enamel, whereas dental papilla is ectomesenchymal component. It forms dentine and pulp. Dental follicle, it forms supporting tissues, namely cementum, periodontal ligament, and the alveolar bone. Along the dental lamina, the ectodermal cells multiply more rapidly and form little knobs that grow under the underlying mesenchyme, each representing the location of one of the 10 mandibular and 10 maxillary deciduous teeth. So as shown in this figure, these are the enamel organs which are formed from the dental lamina, and in both upper, which is maxillary, and mandibular arches, they are 10 in number. That represents the 10 deciduous teeth in individual arches. Here again a histological diagram which shows that this is upper epithelium and this is enamel organ that is emerging from the oral epithelium. Each of these little downgrowths from the dental lamina represents the beginning of the enamel organ of the tooth bud of a deciduous tooth. So these downgrowths from dental lamina, they are then enamel organs. So they represents the beginning of deciduous tooth formation. Not all of these enamel organs start to develop at same time. So all these enamel organs, they don't start developing at same time. They have individual different times when they start developing. So as shown in this diagram, these are the enamel organs of deciduous teeth. As we can see, they are 10 in number because we have 10 deciduous teeth in an arch. And from these enamel organ of deciduous teeth, we can see primordium of permanent teeth. And from the last molars, we can see primordium for the first permanent tooth. On the inside of cap, the ectomesenchymal cells increase in number. The tissue appears more dense than the surrounding mesenchyme, thus represents the beginning of dental papilla. As shown here in the diagram, the upper part is enamel organ and inside it is a dental papilla. So this enamel organs, when it form a cap shape, then the cells inside this, they grow in more in number and they represent the beginning of dental papilla. Surrounding the combined enamel organ and dental papilla, the third part of the tooth bud forms. It is the dental sac or dental follicle, and it consists of ectomesenchymal cells and fibers that surrounds the dental papilla and enamel organ. So here we have seen enamel organ and dental papilla. Surrounding this dental follicle or dental sac is formed. The shape of the enamel organ continues to change. The depression occupied by the dental papilla deepens until the enamel organ assumes a shape resembling a bell. So this enamel organ, its shapes continuously changes, and the depression which is made by dental papilla, it continuously deepens until this enamel organs resemble a shape of a bell. As this development takes place, the dental lamina, which had thus far connected the enamel organ to the oral epithelium, becomes longer and thinner and finally breaks up, and the tooth bud loses its connection with the epithelium of the primitive oral cavity. So as the development is taking place, this dental lamina that is connecting the oral enamel organ and the oral epithelium, it becomes longer, thin, and then it breaks, so in the end this enamel organ, which is a tooth bud, is loses its connection from the oral epithelium. So here as shown in this diagram, the lower portion, which is tooth bud, which is contains enamel organ, dental papilla, and upper is the oral epithelium. These both are connected by this dental lamina, which later on breaks and separates both the tissues. Development of tooth results from interaction of the epithelium derived from the first arch and the ectomesenchymal cells derived from the neural crest. Up to 12 days, the first arch epithelium has the ability to form tooth-like structures. Afterward, the potential is transferred to neural crest cells. Developmental stages. Although tooth development is a continuous process, it is divided into several morphologic stages for descriptive purposes. While the size and shape of individual teeth are different, they pass through similar stages of development. They are named after the shape of the enamel organ, which is epithelial part of the tooth germ: bud stage, cap stage, bell stage, early bell stage, and advanced bell stage. Bud stage. As cell proliferates, each enamel organ increases in size and sinks deeper into the ectomesenchyme, and due to differential growth changes its shape. As it develops, it takes on a shape that resembles a cap with an outer convex surface facing the oral cavity and inner concavity. So here again shown in the diagram, upper is the oral epithelium, lower is the enamel organ, which is in bud stage. The cells of this enamel organ are proliferating and it is increasing in size and changing its shape. Later on, it converts and resembles a shape of cap. That stage will be known as cap stage. Cap stage. As the tooth bud continues to proliferate, it does not expand uniformly into a larger sphere. Instead, unequal growth in different parts of the tooth bud leads to the cap stage, which is characterized by a shallow invagination on the deep surface of the bud. So here, this portion which was in bud stage, it is converted into a cap stage. So we can see an invagination from below. So there is an unequal growth in different parts of this tooth bud, which is leading to a formation of this cap stage, as this tooth bud is appearing like a cap. Bell stage. As the invagination of epithelium deepens and its margin continues to grow, the enamel organ assumes a bell shape. In the bell stage, crown shape is determined. The folding of enamel organ causes different crown shapes and is due to differential rate of mitosis and differences in cell differentiation time. 4 different types of epithelial cells can be distinguished from light microscope examination of the bell stage of the enamel organ. So here this is a diagram which is representing the tooth bud in a bell stage. So here this invagination of the epithelium is deepened, and this is enamel organ is now in a bell shaped shape. Here in this stage, the shape of crown is determined, and as there are different crown shapes, this is because of differential rate of mitosis and differential cell differentiation time, which is responsible for different crown shapes. Advanced bell stage. Advanced bell stage is characterized by the commencement of mineralization and root formation. The formation of dentin occurs after as a layer along the further dentino-enamel junction (DEJ) in the region of further cusp and proceeds pulpally and apically. After the first layer of dentin is formed, the enamel formation starts and proceeds coronally and cervically from DEJ dento-enamel junction towards the surface. This diagram is showing advanced bell stage. In this stage, mineralization and root formation starts. Here, the first a layer of dentine is formed along the dentino-enamel junction, then layer of enamel is formed. Thank you. So we will discuss about these stages, bud stage, cap stage, bell stage, and advanced bell stage in different seminars later on. So till then, have a nice day and enjoy.